All right, great. Can you guys hear me and see my slides okay?
Yes.
Great. So thanks to everyone for coming, and I want to thank Open Boix for this invitation to present my work on this exciting platform. And today I'm going to be talking about how prenatal immune activation leads to a long-term impairment in microglia biology and brain development. And so I'll talk a lot about microglia today, which are the cells that you see pictured on the screen here. These are the primary immune cells of the brain, and they're relatively small cells that tile across the brain in a fairly non-overlapping pattern, as you can see in the lower magnification image on the right. And so since today we'll really focus on microglia, I wanted to first tell you about a few important features of microglia. These, as I mentioned, are the primary immune cell in the brain, and they were first described over 100 years ago by Rio Ortega, as you can see in panel C from one of his original drawings. And these are highly motile cells, both for surveillance functions, as you can see in the movie, in which they're constantly extending and retracting their processes to survey their environment, but they can also have directed chemotactic motility towards an injury or some chemoattractant signal. These cells are also highly phagocytic, as you can see by the lysosomal content in the upper left image. And these cells can phagocytose, synapses or protein aggregates, cellular debris, and even whole cell bodies. And they can digest these material. And they also are very secretory cells in which they can release soluble factors, such as cytokines and chemokines, as well as growth factors and other signaling molecules. Microglia are also very plastic, both in their functions, but also their morphology. As you can see in the 3D image, they have this very complex morphology that can also change based on immune activation or towards a chemotactic or towards a signal that can quickly change their morphology. And it's all these features that allow them to play a really important role in a large number of functions in the brain, both for development like neurogenesis or synaptic remodeling, but also for homeostasis, for things like tissue repair and inflammation or immune responsiveness that I'll talk about a lot today. So these are cells that can quickly respond to an external stimuli, perform numerous types of responses, often with many having cell non-autonomous consequences. Another important feature of microglia is their developmental trajectory. These cells that integrate into the brain very early in development, they infiltrate from the yolk sac around E8 or E9 in mouse. And the medial and lateral ganglionic eminences, as I've shown you in the E13, are some of the first structures to have a substantial accumulation of microglia during the development. While structures like the cortex, especially the upper layers, have a little bit more delayed infiltration of microglia. These cells are also very long-lived and they can maintain their capacity to self-renew throughout their life. So microglia are very strategically positioned in that they arrive early, perform a number of critical tasks for brain development. So if there was any kind of perturbation in these microglia early in development, it could have a long-term consequence on the brain development and function. And this is the idea that really is thought to contribute to the etiology and pathology of neuropsychiatric disorders such as autism and schizophrenia. There's strong epidemiologic data that environmental factors can increase the risk for neuropsychiatric disorders. And these are many different types of stressors can increase the risk, things like psychological stress or malnutrition and infection that I'll talk about today. But all of these are thought to be mediated by some common underlying mechanism that is through this increase in maternal inflammation. On the other hand, there are also immune changes observed in the brains of neuropsychiatric patients, including genetic risk factors and immune pathways, as well as changes in cytokines and chemokines. And people have also looked directly at microglia in the brains of patients with neuropsychiatric disorders. And so first, I want to show you some of the evidence for maternal inflammation and disease etiology. And so some of the earlier studies was from a season of birth study that found a slight increased risk of schizophrenia for patients that were born in spring months, about a 1.1-fold increase in relative risk. And so this led to the hypothesis that maybe gestation during cold and flu season may be responsible for this effect. But that was a very old study, and this has been replicated in more recent studies as well, again, with interestingly the same odds ratio of 1.1, increased risk for schizophrenia in offspring born during winter and spring births. However, the evidence for maternal inflammation is further evidenced by babies born during several influenza pandemics, which caused an even greater increased risk for schizophrenia than the season of birth. Finally, a more direct measure of maternal cytokines, such as IL-6, was found to be elevated in the first half of pregnancy of schizophrenia patients compared to unaffected siblings or control births. And so these findings have been replicated with a number of different types of infections and several different types of patient cohorts. And so this is a really robust association between maternal inflammation and risk for neuropsychiatric disorders. However, as I mentioned before, there's also evidence for immune changes in the pathology of neuropsychiatric disorders. In CSF, for example, cerebral spinal fluid in patients with schizophrenia, I and others have identified elevation of immune cytokines and chemokines in the CSF of patients with schizophrenia. And so this data led to the hypothesis in the field that microglia must be overactive and producing all these extra inflammatory cytokines. So to test this, people from our lab and others use PET imaging of TSPO, or translocator protein, which is an imperfect proxy for microglial activation. It's not just expressed in microglia, it's also expressed in astrocytes and other cells as well. But in patients with recent onset schizophrenia, they found there was actually a decrease in this immune activation marker. So even though these same patients showed an increase in CSF cytokines. So this really threw a wrench into people's understanding of how can microglia exhibit this decreased activation, but have an increase in cytokine expression. Furthermore, numerous studies have also looked at microglia in human postmortem studies, looking at microglia density, for example. And while there were several studies that showed mixed results, this was a more recent meta-analysis that looked across several studies and overall found no change in microglia density. They also looked at gene expression for several microglia genes, and again found there was a decrease in gene expression of several microglia homeostatic genes. So what is going on? What do we really know? So there's reliable evidence for this developmental immune activation increase for disease risk, and there's also really reproducible evidence for the cytokine increase in disease pathology. However, the contribution of microglia, what microglia are doing in disease, has really still been unclear. Furthermore, a major question is if these two phenotypes are at all related, whether the pathological change that we see in patients are at all related to early maternal inflammation. The hypothesis is that the prenatal immune stress induces these later changes, but that really hasn't been tested very clearly. And so what are the changes in microglia that results as an effect of maternal inflammation or prenatal inflammation? And what about the neuronal changes that we see in disease? Are these a result of maternal inflammation, or can they be mediated by microglia directly? And what's the mechanism? So these are the questions that I set out to ask. To do this, I used a translational mouse model based on the human epidemiologic data called maternal immune activation, or MIA, which is what I'll call it throughout the talk, in which pregnant mice get sick during gestation, and then we evaluate the microglia of the brain in the offspring. And so I hypothesized that MIA would lead to a change in microglia, and that these impaired microglia were unable to do their normal tasks, and then would lead to an impairment in neuronal circuit development. So a major function of microglia, as I mentioned, was their immune reactivity to acute activation for things like LPS or lipopolysaccharide, which is the main function that I tested. So in this model, we generate the time-pregnant females and then induce maternal immune activation at embryonic day 9. And then once the offspring grow up, I challenge them with LPS by an IP injection, and then collect the microglia 18 hours later for RNA sequencing. And this is in order to allow the peripheral response to pass so that we're measuring primarily the microglia response. So I have 4 groups that I'm comparing: control and MIA, that are treated with either saline or LPS. So in this first comparison, I'm showing you the difference between the MIA versus control in either the saline group or the LPS group. What we found was that at baseline or in saline, there really weren't that many differentially expressed genes. However, after this acute LPS activation, we see there were many differentially expressed genes, many of them with downregulated. Showing you an example here in TLR1, you can see at baseline, there's not much of a difference between control and MIA, but now in response to LPS, this gene gets induced, and in MIA, we see a reduction in this gene. And this was sort of a trend we saw across lots of different genes. So these data really indicate as the immune activation program that's impaired in MIA microglia. So I next tested how the immune response is different by comparing, saline versus LPS in either the control microglia or the MIA microglia. And so many of the genes show an LPS response in both control and MIA, but we do see a systematic shift in the responsiveness of these cells. And so to quantify this, I'm showing the effect of the LPS response, the fold change in control microglia on the x-axis, and the fold change of the LPS response in MIA microglia on the y-axis, with this y equals x line indicating the genes that would show the same magnitude of the LPS response in both MIA and control. And what we saw was that in the genes that were induced by LPS, we saw about 3 times as many genes fall below this line. And for the genes that were downregulated, we saw about twice as many that fell above this line. So we can fit a line to this distribution of data and show this more shallow slope of 0.71, indicating this systematic blunting of the immune response. So the genes that are induced in MIA, their induction is a little bit less. And for the genes that are suppressed, their suppression is a little bit less. We can also see this in this heat map here, where we have these in this blue section here, genes that are normally not on at baseline. They get induced by the LPS response in the white samples here in control. That's robustly activated, but in MIA, we see this activation is impaired. I next did a comparative pathway analysis where we looked at where the blue pathways are representing blunted pathways in MIA microglia, and the orange pathways are more enhanced gene sets that are more active in MIA microglia. And what we saw is that many of these immune response and viral response pathways were blunted in MIA. However, there were pathways that were more active or more enhanced in MIA and related to mitochondrial function, things like fatty acid metabolism and TCA cycle, while there was a decrease in OXPHOS and redox pathways. So the next thing we did was a single cell RNA sequencing experiment to see if we could identify a sort of disease-specific or a subset of microglia that would just be identified in microglia, where these blunted cells separate from the normal population. And we saw that indeed microglia contributed to all the different activation clusters in response to LPS, although we did see some clusters that seemed to have less MIA cells contributing to those clusters. So to address that, I did an abundance assay in which we looked at the contribution of microglia to each of the clusters in each of the samples and then compared them. And what we saw is that some of these activation clusters, like cluster 2 and cluster 5, for example, showed less of a contribution from MIA microglia to these particular activation clusters. So these data indicate that MIA isn't inducing a particular subset of blunted microglia, but that MIA microglia are contributing less to these specific activation states. So in sum, from this part, we've shown that microglia have this blunted immune reactivity. But next, I wanted to know really how persistent this immune response phenotype was, and if MIA was somehow showing a blunted response to other non-exogenous or non-LPS stimuli. So to address the first question about persistence of the immune response blunting, I performed an in vitro experiment where I cultured microglia either from embryonic day 18, very young microglia, or from the adult MIA microglia. And so for each experiment, we were normalized to control, and then we measured IL-6 and TNF-alpha cytokine in the cell culture supernatant of these cells. And indeed, we found both in the young microglia as well as in the adult microglia, both of them are showing this blunted cytokine secretion. So these data are really indicating that this blunted response is indeed persistent across the life of the animal. So to address the second question about non-exogenous activation, I decided to look at neonatal microglia. And this is a state where microglia are performing many important tasks such as proliferation, migration, phagocytosis. And so this neonatal period is really acting as somewhat of an endogenous activation state. And again, I found many differentially expressed genes in these neonatal microglia at baseline without any exogenous stimulation. And this is really unlike the adult condition where there was no differentially expressed genes at baseline. But we do see a big change in this neonatal pipeline. This is another type of task that the microglia are doing. So I wanted to next see if there was a conserved change between the neonatal response and this LPS response in MIA microglia. So I compare the differentially expressed genes between the two stimuli. And so we see that in genes that were upregulated at P4, we also saw genes that were upregulated in LPS, and these were the ones that were related to this blunted immune response, or ones that fell in this category, for example. And these are related to more homeostatic functions like cell adhesion and cadherin binding and axon development. And meanwhile, for genes that were downregulated at P4 and also downregulated in LPS, these were related to this blunted induction, or the genes that fell in this quadrant here. And these are the ones that were functionally related to the innate immune response and antigen presentation, IRF response genes. So we see that there is a conserved gene expression between the response at P4, this neonatal activation, and the LPS response. Finally, I wanted to determine if the differentially expressed genes in MIA microglia were enriched for genetic risk factors for neurodevelopmental or neuropsychiatric disorders. So I performed a Fisher's exact test to determine if there was an enrichment for GWAS risk factors in these microglia differentially expressed genes. And what I found was that in schizophrenia, there was an enrichment of genetic and risk factors in these microglia differentially expressed genes. So these data suggest that there may be a common underlying pathophysiology between the genetic risk factors as well as these environmental risk factors, providing support for the idea that environmental risk factors may be exacerbating underlying genetic factors because they're impacting the same pathophysiological pathways. So in this part, I provide further evidence for microglia having a blunted phenotype that is persistent across development and is activating or conserved across microglia gene expression pathways. And these may contribute to risk for neuropsychiatric disorders, particularly in schizophrenia. Next, I wanted to determine what's the mechanism for this innate immune response blunting. I hypothesize that this may be due to an epigenetic regulation change, that perhaps the chromatin is in a more closed state, leading to an impairment and activation of the immune response program. So to test this idea, I performed an attack sequencing experiment to evaluate the accessibility of the chromatin. And to my surprise, I found that microglia had actually a more open chromatin conformation. So in this case, the negative fold changes indicate a more closed chromatin structure, and the positive fold changes indicate a more open chromatin structure. So next, to determine how a more open chromatin could lead to a blunted immune response, I performed a transcription factor footprinting assay to determine the transcription factor occupancy sites of these open chromatin regions. Interestingly, we found that MIA had much fewer occupancy sites in the open chromatin, with the top transcription factors being ones that were important for immune responsiveness, such as SMAD3 and STAT1, that had fewer occupancy sites in MIA. So to confirm these findings, I performed a cut and run experiment with IRF1 and STAT2, which is similar to a chip experiment in which I captured the DNA bound to these transcription factors and then performed qPCR for DNA targets. And I found that in MIA microglia in red, they had less capture for target DNA Fas and IL-6, for example, in the IRF1 capture, and decreased DNA content for IL-23 in the STAT2 capture. So these data again confirm that there is less transcription factor occupancy of the DNA in the MIA microglia, which may contribute to this blunted immune reactivity. Therefore, from these data, we identify that microglia immune responsiveness may be due to deficits in transcriptional regulation of this immune response program. But I next wanted to determine if we could rescue this phenotype by replacing these MIA microglia and allowing naive cells to replace them. So to do this, I took a pharmacological approach, which is widely used in the microglia field. I gave a CSF1 receptor antagonist, which is a receptor that's important for microglia survival. So I blocked this receptor between embryonic day 9.5 and E12.5 and was able to successfully eliminate the microglia even after just 3 days of treatment in the embryonic brain. And then once I stopped this treatment at E12, within a week, the microglia quickly returned to their same density. So thus the goal of this experiment was really to eliminate these embryonic microglia that were exposed to the MIA stimulus and allow naive cells to reinfiltrate from the yolk sac after the clearance of this maternal inflammation. Once those animals then grow up, I then evaluated their LPS responsiveness, similar to the assay I showed you at the beginning. And so in MIA, again, we see this more shallow slope indicating this blunted immune response. However, in the repopulated microglia, we see this slope is more similar to control. We can also see this in the heat map, where the red genes are ones that are induced by the LPS response, and the blue genes are ones that are suppressed in response to LPS. And we can see in MIA, we have this blunted responsiveness, but in the repopulated microglia, we see an amelioration of this immune responsiveness, indicating that indeed the repopulated microglia are able to have a more normal immune responsiveness. Similarly, in the single-cell RNA sequencing experiment, we found that, the repopulated microglia exhibited a comparable profile to the endogenous microglia. They contributed to the same clusters. And more importantly, the distribution of those activation states, which we had seen were reduced in the MIA microglia, have now been restored in these repopulated microglia that are now contributing to the different activation states more evenly, or more similarly to control. Thus, these repopulated microglia were able to restore their immune reactivity. But the final part I wanted to ask about were the non-cell autonomous impact of MIA microglia. Everything I've shown you so far has been about microglia, but what about the effect of MIA and microglia's function on other cells? So I looked at protein expression to further validate the RNA sequencing results. And so as you can see, we in the MIA in red, in response to LPS, we had a decrease in the protein expression of TNF alpha that was again ameliorated in the repopulated microglia. But in these same animals, I also looked at IL-6 expression in the astrocyte fraction, and I found that astrocytes had a slight elevation in IL-6 in these MIA. That was also ameliorated by these repopulated microglia. Thus, even though the microglia have a blunted immune response, astrocytes may be compensating for this blunted response by increasing their cytokine expression, or microglia negative feedback may also be impaired, leading to an increase in the signal from astrocytes. Next, I looked at neuronal activity. I looked in the striatum because we found that the blunted immune response was greater in the striatum. And so majority of the neurons in the striatum are split into these two classes, a dopamine receptor type 1 and dopamine receptor type 2 medium spiny neurons. Thus, we can record from either these D1 or D2 medium spiny neurons. And what I found was that in the D2 specific medium spiny neurons, we showed this increase in the paired pulse ratio. It indicated that there was a decrease in the presynaptic probability of glutamate release in the circuit. To ensure that this was not a result of decreased excitability in the presynaptic cell, we also measured the mini EPSC frequency in the D1 and D2 cells. And we found that specifically, again, in the D2 medium spiny neurons, there was a decrease in this frequency. Now, in the rescue, we wanted to see if microglia were at all contributing to this phenotype. We again see this decrease in mini EPSC frequency in the MIA mice that is rescued by the repopulated microglia. And so these data really indicate that MIA microglia have a causal role in the impaired connectivity of this D2 circuit. So in conclusion, these data indicate that MIA leads to a blunted in the responsiveness of these MIA microglia to acute LPS activation in the adult. And we also see this intrinsic blunting of the intrinsic activation from these neonatal stimulus, leading to changes in conserved gene function pathways, including things like the immune responsiveness. So these data really suggest that MIA may be acting as a tolerizing stimulus to induce this sort of innate immune memory in MIA microglia. And while we were expecting a priming phenotype or microglia to be hypersensitive, what we found was just the opposite. And I think that this could be as a protective adaptation for microglia. When microglia are being born into a brain with a heightened immune environment, it may be decreasing their set point or their threshold to avoid being hyperactive to a subsequent immune challenge. And this mechanism may be mediated by epigenetic reprogramming. And this is something that has also been observed in metabolism or high-fat diet during pregnancy as well. We've seen similar changes in the shifts in their adaptation based on what the prenatal environment looks like. We also observed strong parallels with the clinical data, which was really exciting to us. So similar to the PET data, we also observed this blunted in microglia reactivity. So that was encouraging to see in the animal model what we also observe in patients. We also saw the increase in IL-6 from astrocytes, which maybe gave us just a glimpse into what we may be going on with the CSF results, and that although microglia may be less active, an increase in cytokine signal could be coming from other cells. And in patients, it's important then to continue to look at other cells like astrocytes or even endothelial cells that could be contributing to this immune signal that we observe in patients. We also saw that these microglia were causally linked to a circuit that's important in neuropsychiatric disorders such as schizophrenia, namely this D2 medium spiny neuron circuit that's known to be perturbed in disease and is a major target of antipsychotic medication. So in sum, these results really provide an insight into what's going on in microglia and disease and to really clarify some of these clinical results that we've seen. So I want to thank my mentor, Kirasawa, and a number of students and colleagues and collaborators that have worked on this project with me that have now moved on to better things in grad schools and medical schools or in new jobs. And so their support has been entirely influential and very helpful in this study. So, and with that, I'm happy to take any questions.
Thank you very much for a very, very interesting talk. As you said, now it's time for questions. Indeed, I have one. Did you check also the behavior of the mice, like if there is any more behavior that they have a different behavior in the MIA treated ones respect with the others?
Yes, so we did see some behavioral changes. So behavior has been, sorry, my screen is like gone away. I don't know where you guys all went somewhere. Anyways, that's okay. Maybe I'll just stop sharing. Okay, there we go. Now I can see you guys. Yeah, so for the behavior, we did do behavioral tests. We saw changes in things like social interaction and amphetamine challenge, were two of the ones that I had tested. We see a hyperlocomotion in response to amphetamine and a decrease in social memory recognition. But also these MIA, this model has been highly characterized in the field in general to have several different behavioral domains that are impaired. So we really focused on the microglial phenotypes more than the behavioral phenotypes.
Okay, thank you. Now I think Robert will ask you a question.
Hi, what a whirlwind, wonderful talk. Thank you so much for sharing that with us. I have and I'm at UAB. Oh,
great.
Just so you know where I'm coming from. I was interested kind of towards the end of your talk, you had referenced that these are, concordant findings with what you see in maternal high-fat diet. I'm quite interested in metabolic syndrome and the whole cluster. And it made me think about your model, the LPS challenge. I was curious, two things about it. One, I was thinking LPS challenge often induces hypotension. It can be profound hypotension. And I was noticing one of the differentially regulated genes in your microglia, it was a volcano plot you had on one of your many wonderful slides, was for RAMP2, which is a co-receptor for one of the most potent vasodilators ever identified. And so I guess I'm just wondering, as you chew through these things, what do you see, what do you think about this relationship, between metabolic disease risk and the way that these inflammatory challenges can kind of be metabolic challenges or physiologic challenges as well? I'm thinking about the hypotension. I know that's kind of a convoluted question, but I just want to hear what you think.
Yeah, no, that's super interesting, actually. And I'm very interested in that because actually ACE, angiotensin converting enzyme, is something that's come up as a risk factor for schizophrenia. It's also come up in Alzheimer's disease. So I think there's definitely a link between those types of mechanisms and what we see in the microglia. Mechanistically, it's very unclear, like ACE is not expressed in microglia. So that's going to be something that's happening in parallel. But yeah, I think that's something that's very interesting. I mean, we've just looked at microglia. I didn't look at things like endothelial cells. I just know from the literature, high fat diet and the way that the offspring metabolize, fats becomes very different. This is an adaptation that's occurring. And so, when we were very confused by what these results could mean, that was sort of some of the literature that I was drawing off of to be like, how does this make sense? And it was like, okay, when you look at these other models, this isn't a suppressing phenotype. This is maybe what we should have been expecting at the beginning. So thank you. Great question.
Thank you.
We also have a question. Yeah. And now we also have a question from Americ.
Yeah, I wanted to ask you, did you check IDGAX gene? Because it looks like through the pathway that you showed that are upregulated in your MIA microglia, fatty acid metabolism, that it looks like the CD11C positive dendritic microglia from Hajmeyer and Wojtarczyk, and they suggested a role in myelination, but it was never like really reproduced highly, and we don't understand really how they act on myelination. And could it be that they last longer because the CD11C positive microglia, as you will see later, appears and disappear? The program disappear, but do you have any other genes like IDGAX, CLEC7A that are more expressed in your MIA microglia?
I'd have to go back and look specifically. I can't remember off the top of my head. But when do the CD11C cells show up? Do you know when they first, are they part of the early wave or are they a later wave?
So they are later waves. They arrive slightly later, but before birth, and they stay after birth for 2 weeks, and then they disappear.
So I think what we're looking at in the adult is definitely the microglia, just based on, where this is happening. We're evaluating cells much later than from the insult. So the time of insult and later, and the microglia were, this months apart. The other thing is that we're using a really early MIA model, so E9. So there's several different camps of MIA. So people that do an E12 model, there's E late, like E17, E15 models. So we're definitely hitting the very first wave of microglia that come in. So that's sort of one of the reasons I bring up the MGE and the LGE phenotype, because I think, we see a phenotype higher in striatum. We're hitting them very early. Some of the earliest microglia end up in the MGE and the LGE. They go on to develop into the striatum and the cortical interneurons, which is also impaired in these MIA models, as well as in schizophrenia. So I think that the timing of the immune insult and when you're seeing these stressors is definitely going to impact particularly what microglia may be susceptible. And that could all be as a result of the timing of this insult and what microglia are susceptible. But what mechanisms are controlling microglia susceptibility windows is something that's completely unclear and something I'm very interested in looking at in the future.
Thanks.
Thank you.
And we also have a question from Chi Chung Lin. She is asking, you show less transcription factor occupancy in MIA microglia. Do they show lower expression of transcription factors?
Yeah, so it's a little bit of a chicken and egg question. For STAT1, for example, we see less STAT1 gene expression. We also see less occupancy on the chromatin, and then we see less target genes of STAT1 that are also decreased in their gene expression. So it's possible that the reason we see less transcription factor occupancy is because there's less expression of those transcription factors there. So it's a little bit of a chicken and egg that we don't have the exact, timeline of the mechanism, which, we're just looking at 18 hours, which is kind of late for the LPS response. And so I think that, if we were to look at more of a longitudinal trajectory, we might be able to capture some of these things more clearly.
I see. And there's another question now from Octavio Caspe. Did you test microglia rescue through anti-inflammatory cytokines?
Yeah, so I showed that in sort of the end where we looked at the TNF alpha protein expression, for example, from the rescues was normalized back to the control levels.